Screen share. Well, good evening, everybody. Thanks for joining. Sharing screen. Presentation mode. Presentation mode. There we go. So my task is to introduce you to anemones, fascinating creatures. The Greek word refers to a wind flower because, as anyone who has seen them, they're just wafting there. It looks like they're blowing in the wind as they're being moved around by the current. Absolutely gorgeous animals. Most of the photographs that I'm going to show you now are taken with one of these two point and shoot cameras, very cheap. The Nikon, I think, is no longer available. The Olympus is still available in its latest versions. One or two of the photographs have been taken with a bigger camera in latter years. But the reason why I first bought a camera was because I had great difficulty trying to describe when I came back from a dive, asking Tony or some other knowledgeable person, what was that yoke with the thingy-majiggy? And rather than try and describe it, I discovered that if I took photographs, I would be much better able to identify what we were looking at. So sea anemones are Cnidaria. They are related to corals, hydroids, and jellies. The thing that they all have in common is that they are weaponized with stinging cells called nematocysts. And these are variously used to poison or cut or entangle or barb an enemy or a potential food source. And that's why we have these stinging animals underwater. Their anatomy is very primitive. There's no circulation, but they are able to pump themselves up with water. Inside the sea anemone, there are different septa which divide them into different compartments, and they can, as you can see here with this example of a dahlia, which is quite large at the moment, the tentacles will trap and prey and feed it into the mouth, which also acts as the anus. So the most unfortunate creature on the planet has to take in the food and get rid of it through the same orifice. So when it comes to describing the anemones, we refer to several aspects of the anatomy. And the first, of course, is the mouth, circled here in the blue oval. And then you have the oral disc. Now, some people will refer to the entire top of the anemone, which includes the tentacles, as the oral disc. But the tentacles then are the third thing to look for in terms of characteristics when you want to come to identify them. And in particular, you'll be looking at the numbers, their length, their pattern, and their colors. But that's not going to tell you an awful lot about the anemone, because the big question is what is underneath. And you're looking at, you've got a bird's eye view of this anemone, but you cannot see the column and you cannot see the base, and therefore it can be more difficult to identify accurately. So here's a bird's eye view of a big pillar in Berlin, I think it is. And you can't tell very much from it. And just imagine that you're swimming over an anemone, you're looking down at the top of it. But if you then come down to the side of the pillar, you'll be able to see all the different characteristics that help you to classify it according to its type. And that's the importance of looking for the column in the anemone when you're coming to try and identify it. So in terms of their behaviors, there are several well-known anemone symbioses. The first, of course, is our favorite Nemo, who hides in the anemone. He and the anemone then have mutual benefit to each other. This is a clownfish variety taken in Indonesia, and that's their big anemone home there. This is another example of lovely symbiosis where you have a clownfish being treated by a cleaner shrimp, and the shrimp is serving the clownfish is serving the anemone is serving them both. So real symbiosis goes on between anemones and other species. Closer to home, we have symbiosis with snakelocks. So in their natural state, they have this rather dull, beigey, grey sort of color, not terribly pretty. And by the way, the snake's locks anemones cannot, tentacles, sorry, cannot retract. But when they are colonized with symbiotic algae, then they take on these beautiful colors, the green and the purple there. And again, you have symbiosis here because the algae are photosynthesizing and they're providing energy to the anemone. Another symbiosis that's quite common here, although we haven't seen it much of late, is the little spider crab, Inachus, in snakelocks anemones. Now, there are some dive sites where they're definitely still in symbiosis, but in New Quay, for example, we see them sitting on jewel anemones or whatever else happens to be around. But that's another symbiosis to keep an eye out for. So when you're swimming over and you see a lot of snake locks, have a look for the little spider crab that will be living in very close association with it. And then the other symbiosis that we do see here from time to time is the Adamsia palliata, the cloak anemone on a hermit crab. So the sea anemones are divided into several little groups here. Lots of them occur as individuals attached to a hard surface, and there are over 1,000 species, we reckon. Some of them are burrowing anemones, so you'll see the top of the animal, but most of it is buried underneath the sediment. And then you have encrusting anemones, where the individuals have a common base, and we'll show you examples of that. And then we have the knobbed anemones. So in terms of individual anemones, this is one that will be recognized by all divers, the plumose anemone. It's our tallest one. It comes in various different colors, white, orange, brown, green. And contrary to what I said at the beginning, I haven't shown you a column, but when you take a step back and you can see then that these are quite tall, you'll often see them in big aggregations like this. And they happen to be all the same color, but you'll often get them as mixed colors side by side as well. They have that beautiful flowery appearance, which gives them their name, the anemones of windflower sort of appearance. Another very common anemone that we see here is the dahlia, Urticina fellinea. It's one of our larger anemones. It has 160 short, stout tentacles, and you can count them if you like. They're all there. It gets up to 15 centimeters across at the base, and as I say, one of the larger anemones that we see here. Now here it is. It's with hydrostatic pressure. It's pumped itself up with its own sort of surrounding seawater, and you can see then that there are these little verrucae, these little warty structures on the column, and that it's quite sticky. So you'll get little bits of gravel and fragmented shell that stick to the column. They come in a variety of beautiful colors. It's almost endless, the different patterns that we can see in them. And they've often reminded me of, county football jerseys because there's so many of them. They have an amazing appetite. So this is a retracted dahlia, and that's the tail end. Well, actually, it's the claw, the claw of a velvet swimming crab. And you can see that the whole animal has been engulfed. You can see also very clearly here all the wart-like structures, the little verrucae, those little white knobs on the column. But imagine the strength of the digestive enzymes that are in that inside that animal that can take on a crab and digest it. Here's another crab that came to a sad end with a dahlia. And here is the tail end of a lobster. Now, judging by the color of that shell, I reckon that it was discarded from Lenan's Lobster Bar in New Quay, sort of cooked and then eaten, and the rest discarded, and it ended up there being recycled in this dahlia anemone. Here's a worm, polychaete, that stuck its nose in where it shouldn't and was not able to retrieve itself. I spent a couple of minutes just watching to see what would happen if he'd managed to free itself, but it just couldn't. It was stuck, and that was going to be, it was a meal, and that was the end of that. Here is a piece of kelp with bryozoan, and that's been consumed by the dahlia. And finally, I don't know what that is, but that's also been consumed. It looks like a spicule of some sort, or a remnant of something or other. I just couldn't make it out. It's quite cartilaginous. It feels quite stiff when you put your finger on it. So they have an amazing appetite. The next group of anemones to consider is the elegant anemones. We used to call these Sargathia. Now they're called Silista elegans. Interestingly, they have a very strong adhesion to the hard surface underneath, almost concrete-like is the secretion that holds them down. The base is broader than the column, and it has these numerous white suckers on its column as well. The small pores that are said to be just visible as dark spots on the upper part of the column, I have never seen. I wonder if I would have to take a microscope out to see them, but that's what the description says in the textbook. They come in 5 different varieties. Here's the sort of orangey brown one, the miniata. And then there is a rosea variety, which I think are absolutely gorgeous. This particular animal posed like that. I didn't poke it. I didn't Photoshop it. It just appeared, and it became the Facebook image for St. Valentine's Day, whatever year that was. And then there's the white one, the Nivea. And then there is the Venusta. That's the 4th one, which is quite yellow. These are all the same animal, just different varieties. You can see that their mouth and their discs are very similar in terms of shape, identical. And it's just the colors that are changing here each time. And then the 5th variety is the Aurantic Yaka, which is a little bit duller, but said to be quite distinct with those orange tentacles, gray disc, and then that lovely pink color around the outer edge. We've been very fortunate because we've seen all 5 varieties of the Silista elegans in one dive site, although this particular one is the rarest by far. When I come across fields of these, I think of a certain poet, wandering lonely as a cloud, and you just see these fields of beautiful, elegant anemones. They will also occur like this in clumps on vertical rock faces. And the close association with sponges as well. They're quite versatile in that regard. And the rosea variety tend to be much less numerous, and they seem to like to have more space for some reason. And that might just be the dive site that I've seen them at, so they might be different elsewhere. And again, here they are occupying a sponge, but you can see that they're quite widely spaced. Now, interestingly, we saw these catch tentacles, so-called, and wondered what purpose they might serve. And the idea, the different thoughts about the catch tentacles is that they might be used to fend off neighbors, especially anemones of a different variety, because some anemones they think do have catch tentacles for that very purpose. We wondered whether they were reaching out to find a buddy so that they could reproduce, but we're just guessing. Don't really know what those catch tentacles are for. The catch tentacles that are used to sting neighboring or encroaching anemones are said to come from the base of the animal, but I think these tentacles are coming from the top rather than the base of the animal. So the jury is still out on that. It's not likely to be reproduction because anemones will have these stacks of coin gonads inside, male and female, and they will release the gametes into the water column for external fertilization. But they do say that different types of reproduction can occur in different anemones. So I got very excited when I came across this miniata variety because I thought that looks to me just like mitosis in cells, biology 101, how the nucleus divides up into 2 and goes off in 2 different directions and one cell becomes 2. So I got very excited about that, but then I was deflated when an expert told me, no, that's just a Siamese twin. That can happen from time to time. It's just a Siamese twin. So people don't really know quite yet how they reproduce. They have some idea, but not all of the idea. So another individual anemone stuck onto a hard surface is the imperial anemone. And this is a very, very different appearance. As you can see, it has these very short um, stalks and terminal knobs, which have a much more clumpy looking appearance. There are up to 150 knobbed tentacles in 4 different cycles and a very broad base of up to 9 centimeters wide. I think I have another one of those. Yeah, there's another good example of the short, stumpy tentacles and arranged in very tight circles. Another standalone anemone is the clock face anemone, Pichia cylindrica. And the characteristic feature here is the concula, which is the little 3-lobed funny mouthpiece there in the very center of the animal. So that's the individuals attached to hard surface anemones. And now we look at a few of the burrowing anemone-like or tube anemones. And this is one that you'll see a good bit out of in Connemara here, in Rossrow and other places out on our side of the country, the Cheiranthus lloydii. The tube is going deep into the soft sediment, and then you've got these different rows of tentacles. You can see there in the very middle bit there are very short tentacles, and they're surrounded by much larger tentacles on the outside. The color here is straight white, but it can occur as brown tentacles as well. By the way, I should say that I'm not a marine biologist. There are a few marine biologists on site tonight, so if I get anything wrong on that side of it, please be sure to correct me. Another very beautiful animal is the firework anemone, Pachycheiranthus multiplicatus. Quite rare. We have them here in Kilkieran Bay, and they occur as well in Kenmare and in the west of Scotland. They are absolutely beautiful. In Ailbui, we have like a whole field of them, and it's like going down to watch a ballet in full swing as they waft around with the current. Taking photographs of them is extremely difficult because there is so much sediment floating in the water, and there's often a little bit of a current as well with it. So, and it just takes one flip of your dive buddy's fin to create a cloud of muck that's impossible to photograph through. So, if you can get any clarity at all on these photographs, that you're doing very well. Now you can see here that the tentacles in the center are shorter and they have a sort of a mauve appearance, surrounded by much longer white and very elegant tentacles. But they can also occur as banded tentacles like this animal here. So they're absolutely gorgeous. There's the tube in the firework anemone, they reckon, is about 1 meter into the soft sediment. So all we're seeing is the very top of the animal, and the vast majority of it is underground. And now we'll come to some of the encrusting anemones. These are colonial, like this white cluster anemone, Parazoanthus angiocumus or something of that nature. It crusted on a hard surface. So what I would do in a situation like this is I zoom in because I love to have the contrast of the elegant reaching for the sky tentacles. You can see there that there are two rows. There's the inner circle of tentacles that are going straight up into the water column, and they're surrounded by another circle of tentacles that are coming out towards us here. They're on the horizontal plane. So those two circles of tentacles. Now this is encrusting. So the idea here is that the individual animals share a common base. But because I zoom in to get what I consider to be a pleasing image, my dive buddy Tony gets a better image to show that the bases are actually joining together. If you look at some of those, you'll see that the bases are coming together, and so they're called colonial anemones. And then in terms of the other types, then we have the knobbed anemones, Coronactis viridis, or the jewel anemone, which is absolutely, again, absolutely gorgeous. Up to 100 knobbed tentacles on each tiny animal in a variety of colors. You can see there the very clear pink knobbed looking appearance. The name viridis comes from that beautiful bright lime green sort of color that you can see on this animal. And again, you can see here the very thin line of bright green. That's what's giving it its name. Now, when these are retracted, they have a very smooth column. And there's another example of how smooth the columns are. Some of my diving friends tell me that we should be looking out for little amphipods or little insect, can't remember what you call them, amphipods of some sort that are often found in conjunction with these anemones, because apparently that happens elsewhere, but I've never actually seen an example of that as yet. They love to hang out on vertical rock faces. All the different colors can be coming together, but they can also find little nooks and crannies, such as this kelp holdfast, and those anemones were thriving in hiding in the holdfast of a kelp in the middle of the channel. They were absolutely beautiful. Again, the very clear viridis color there. And as I say, the variety of color is almost endless. Oh, that's the same photograph. Sorry about that. Now, the Devonshire cup coral is not an anemone. It is an anemone-like in that it has tentacles and they are knobbed, but it is a solitary coral in a hard cup. So it's a different animal entirely. So with anemones like that, you know, who needs friends, right? But if I get lost on a dive, my dive buddies will come back to the to the last clump of jewels, and they invariably find me there, mesmerized by the beauty that we're looking at. But I do want to give a shout out to one friend, that's Tony, who's working so hard for Sea Search and has done so much for all of us in terms of increasing our knowledge and organizing workshops and making sure we're getting out diving and recording our different species and all of that. So well done to you, Tony. Thank you so much for all you've done for us. I'd like to invite everyone to join this group. Not that it's mine, but it's a very useful group. Because what you can do is when you come across something that you don't recognize, like for example, I came across this in the middle of a field of elegant anemones, and I wondered if it was a juvenile elegant, if it was maybe a baby that didn't have yet or the characteristics that it would have as an adult. So what I was able to do was go onto that website and ask any idea what this could be. Is it a juvenile elegant, or, you know, what else might it be? But the answer comes back very, very quickly from somebody who knows what they're talking about. It's Anthoplura species. And you can see there, Dawn Watson came in quickly with that. Julian unfollowed up and agreed. And then I can read up on that particular species of anemone and begin to learn to recognize it myself. Um, Now, you would think that having said everything that I said and coming back to this original image, what's that? You see, it doesn't fit into any neat category. It is actually an elegant anemone, Celesta. But we had a big discussion trying to figure out, was it really that, or was it a funny dahlia, or what was it? But it does not fit into any of the 5 different species that I'd mentioned in relation to the Celesta, the elegant anemone. And in discussion with Bernard Picton, he would say, well, he's not really that keen on the distinction of the 5 different varieties because he thinks that there are mixtures just like this one, which prove his point. So it's sometimes things are just not going to fit into the neat categories that I have just described. And here's another example that I think illustrates that. This is something that Bernard himself posted, and you can see the name there in the top right-hand corner, an actinotho spirosida or something of that nature. And I thought that looks just like one of our Nivea elegant anemones. But if you look at, you can see the difference now. If you look at the column of this animal from here, you can see that, for example, the base is not broader than the column itself, that it's much taller than it is broad. And you can see here that the tentacles are quite different from the elegant anemone tentacles. So those two characteristics do help to point you in the right direction. Now, if you're still, you'll still get caught out. So here's another example of an anemone that I found in New Quay, and I didn't know what it was. If you look at the book for this particular species, you say, okay, well, if they're supposed to be red and blue spots and all the rest of it, and this is the Actina equina, but you can see here that they say that each anemone is a single color and the column is smooth. I just need to move this so I can see what I'm looking at here. And not patterned. Now that's according to one book, but then look, there is a pattern here. It's streaked on the column. It's got these brown and yellow lines all the way down. So then another, the habitats.org.uk website does speak about streaks on the column. So even when we read books, we might find some conflicting information, which can leave you feeling frustrated. It certainly can leave me feeling frustrated. But the giveaway here for the Actinia equina is the blue dots that are around the mouth. Here we go. And so here's another very useful resource for identifying different species. You can go into your species and then look for all your different anemones, and you will find photographs for most of them and clear descriptions as well on what to look for when you're trying to identify them. So there you are. Thank you, Tony. I'll hand back to you. How do I do that?
You can just unshare, and then John can just share.
I have to say okay to the recording. Stop share. There we go.
Yeah, thank you very much, Joe. That was excellent. I forgot to say when we're starting that if anybody any questions, just stick them in the chat, and we can bring them up as talk goes on, or we can hit them at the end. So if you have any questions, just stick them in there. I don't see any questions there at the moment, so I'll hand over to John and let him tell us all about fishes.
Okay. It's me to speak, is it?
Yes.
Okay. Thanks, Tony. I'm a zoologist. I study ants and bees and not really fishes. So if I say things wrongly, please, you can fight with me afterwards. I do say that as a zoologist, I do say fishes when I'm meaning different fishes. And I say ducks when I mean different ducks, as distinct from fish and duck. But anyway, other than that, that's my use of English. And I think it's the traditional zoology use of English. And I said I would say a little bit, I was really picking up on last week's presentation by Martin, and that basically you need a camera and lighting and a compatible dive body. And you've just been listening to one of them, and the person introducing our host is another one of my compatible dive bodies. So we go for long, slow, unrushed dives, not quite 2 hours, but almost, sometimes in UK. That's my setup. You can look at it later because people have asked me. I'm still using a 7-year-old TG4 Olympus. And I mainly photograph. I don't have a flash. I have a video torch. And it's now the TG7. And they're the settings I use. And if anybody wants to contact me, I can talk them through them if they're interested in those. I thought I would say this because Martin was mostly talking about using heavy gear SLR camera. And I have that when I'm photographing ants and bees on land, but not underwater. And so after that, I'm talking to you about fish or fishes. And I think, first of all, one tells you about the book. And the book for inshore fishes is by Lynn Baldock and Francis Dipper. And both of them have, and Lynn especially, has been diving a lot in Ireland. So it's quite accurate from the point of view of saying what species you're likely to come across. It's also quite detailed in places. I just want to prepare you a little bit for it. Also, I will say that these are the sort of books that will probably be printed just once. And once they go out of print, they go out of print. Hopefully, enough said. My advice on more or less any group of animals, whether it be ants or bees or underwater, is to take on one group, learn their features. So spend the night before and then pretend you're seeing nothing else except them on the next dive. If you can, and if you have a camera, take some shots, even out of focus, to show the critical features. And effectively, what I'm saying is that you need to know the critical features rather than necessarily remember them all underwater. And certainly some of the things I'll be saying later are things that you won't be recognizing underwater. You'll need a shot to be sure what you saw. So just for the sake of completeness and to satisfy my zoological background, I'll start with cartilaginous fishes, and I'm only just going to show you a few. It comes as a shock to me in later life that these are now called cat sharks. I would have learned these as dogfish, and it's the small spotted dogfish. But these have cartilage skeleton rather than a bony skeleton. And effectively, if you take one of these and steamroller it in evolutionary time, the gills go underside, open on the underside, and you get the skates and the thorn. The skates and rays. And this is a fish that I've not seen. It's the electric ray, and it was seen by this one was seen by the UL diving club some years ago in Kerry. So I'm just saying that what most of us call dogfish, the book is, they're now cat sharks. And we're talking about the small spotted cat shark. Now, the vast majority of fishes have true bone, and they have gills under a gill cover called the operculum. So you do need to get to know and try to observe a few things underwater. Can you see a dorsal fin? Can you see an anal fin, the pelvic fin? Now, in this case, in the earlier fishes, the pelvic fin was back where the pelvis is, and the pectoral fin is where you'd expect it, up in the front of the fish. But in more advanced fishes, the pectoral fin has moved up, and the pelvic fin, they're now calling it here at the ventral fin, is moved underneath the pectoral fin. And sort of things that you can also notice underwater are how many dorsal fins do you see? So if you see 3 parts to the dorsal fin, then you're looking at the family of the cod family. And then does it have a barbel? And finally, along the middle, try to see the lateral line. And is it straight or is it arched? Or does it have a bump on it? And those sort of things can help. Try to get the approximate size. So hold up your hand towards it. Is it your hand length, your elbow, hand to elbow? Is it your arm length? Any obvious black or coloured or white spots? The lateral line I've mentioned. And then the arrangement of the fins. Does it have a barbel? So this is a pouting and nuki. And again, barbel, there's the lateral line. There's the lateral line. 3 parts to the dorsal fin. So it's a cod family. And we see these regularly in Newquay, August, September time. These are young babies. I usually tell people to learn one group at a time, and I would always suggest starting with the wrasses, because in some places, if you dive in out of Kilkee, the dive at George's Head, you will see all five species on the same dive. So one of the species has very different males and females. So this is the male cuckoo wrasse. This is the female. She has black and white along here. If you want to get into some pornography, Google about sex changes in cuckoo wrasse, or in more or less all of the wrasse family. They all start off as females. The male has a harem of females. If a male dies, or if they're looking for another male, one of the females becomes a male. And on some dives, you'll see these. We better not mention this to Donald Trump. Females becoming males. One of the smallest of the group is the rock cook, which is quite common. You tend to not see one, you tend to see about 10 or 12. And they have these little black lines towards the tail, and they tend to have blue lines. This is a fairly brightly colored one, blue lines on the face. The gold cine wrasse has the spot on the top of the base of the tail. It's slightly bigger than the rock cook. And then the corkwing wrasse has the spot in the middle of the tail. But quite often you don't see this spot, but I think the spot on the previous one is reliably there. So if it's not there, you should begin to know what corkwing wrasses look like anyway. And then all of the rest tend to be ballan wrasse, and they're big slobs of fish. And there's quite a variety of colours. And then there's one that we're supposed to be looking out for. And it has pink mouth here, and it has a dark spot behind the operculum. This is the operculum, the gill cover. Flatfish. Flatfish can be right-handed or left-handed. And if you haven't been through this before, I suggest that you start holding your hand in front of you, and your left hand and right hand, and you're trying to imagine the mouth and the operculum. And now put that down on the ground, on the table. And the right-handed fish are plaice and flounder, lemon sole. And the left-handed fish generally are top knots, because effectively that's what we tend to see on dives. So now, if you were trying to have your hand laid out on your table, I'm trying to do this as I speak to you, the operculum, which is here, and the mouth, which is here, really only fits on your right hand. So that's a right-handed fish. And there's the mouth, there's the operculum. Could I ask, are people seeing my mouse arrow? Yes. Okay, thanks, Joe. Okay, so that's a right-handed fish. And this one is also right-handed, so it's probably a lemon sole, but it fits, and the slight bump in the lateral line just here also fits the photograph in the book I told you about. And how many can you get into the one photograph of these? So that's just at the bottom of the steps in Newquay. Left-handed fish. Again, now you have to hold out your left hand, and the mouth is here, and the operculum is here. And we see these under the ledges in Newquay. And when you start going back to the book, you check out, and the common top knot has the dorsal fin. This is the mouth, so this is the dorsal fin coming as far as the mouth. And that's the diagnostic feature. There's the fin coming along, and there's the mouth. And if you're diving out of out of Kilkee, for example, and this is the Blue Pool dive, you sometimes see these. They're usually upside down in small ledges and quite brightly colored, pink spots and everything, but it's a very small fish, smaller than the size of your hand. And in this case, there's a space between, the mouth is here, and there's a space between here and the fin. And so that makes it a Norway top knot, a Norwegian top knot. Blennies and gobies. Effectively, gobies have the dorsal fin split into two parts. Blennies have a single dorsal fin. So the single dorsal fin, starting with the blennies. This is the common tompot blenny. Everybody photographs them. But then, especially out of on the west coast rocky shore dives, rocky dives, you get the red blenny, which has these red marks here. It obviously looks much more red, and the male in breeding has this blue spot. This is probably a female. And then we're into gobies, and the gobies have this split between the first two parts of the fin, and the easy one to identify is the leopard goby. And now I'm beginning to show you that if you want to try to start identifying your gobies properly, the good photographers are telling you to focus on the eye, but you're trying to focus on the top of the pectoral fin. And hopefully you can see here the free filaments. And if you start counting these, there are 5, and that makes it a rock goby, even though it looks quite black. And the black goby that I showed you, when you zoom in, there's the eye, and you can just about see that there are 3. It's a bit not as, it's more focused on the eye than on the filaments, but there aren't 5. So it's either 2 or 3, and this one is a black goby. I haven't done the same tricks on this one, so I'm not going to say which it is. This one is swimming in the water rather than sitting on the bottom, and these are quite common, and you'll see the 2-spotted goby. This one means traveling to see it, but they're common in Lochine, the red-mouthed goby, and they come across them. I've come across them. I've not got a good photograph in Salt Lake, but I've seen enough to know that I've seen them there. And then looking at other small fish, this is the long-spined sea scorpion, and very, very good at camouflage, but the diagnostic feature, and you can always see it in the field, is this little white barbel, I think, on the side of the jaw. White barbel hits you in the face, so there's the fish. I like this one from the point of view of camouflage, but the white barbel gives it away. And the purple is part of the fish. The purple here is part of the calcareous algae in the background. And this one doesn't have the barbel, and it's the short-spined sea scorpion fish, and they can be much bigger, but they can also be just the same size as the other ones. This is a fish that I began to see in my early years of diving, long before cameras, in at the back of ledges, overhangs. And eventually I got to take some photographs of the tadpole fish. It's sort of purplish, disappears once you start looking at it, doesn't like being seen. Then we have sticklebacks and pipefish. They are related to seahorses, so look at their heads. Now you want to go to, Joe mentioned, and Tony is an advocate of diving in places that most people don't go diving. Zostra beds, eelgrass beds. They're sort of dives that quite often you can stand up if you run out of air. Because that's the depth you're diving at, and you're unlikely to have run out of air. But I'm talking in terms of 2 or 3 meter dives, and you look in the eelgrass, or you look at around the holdfasts of kelps, and you just keep staring. You need patience. And these are sticklebacks in this type of habitat. Notice that the head looks a little bit like a seahorse, and in the case of the sticklebacks, they have the larger tail. And then we get into the pipefish themselves. This is the greater pipefish with a very much seahorse type head and very fancy colouring. And this is the type of habitat that I'm talking about on eelgrass zostra beds. This is at Glenina Pier near Ballyvaughan. And if you stare at this long enough, you'll see nice little pipefish like this. It's some years ago now. We haven't had them in recent years, but one hears about triggerfish being seen on dives around the country. And I keep telling people that I wouldn't trust a triggerfish with my little finger. I think the Mediterranean ones certainly have chopped off a few tops of fingers. Just be careful, but they're called triggerfish because they have a front spine here, which they use to jam themselves into underhangs like this. And we have seen juvenile gurnard at Newquay. They really take you for a long, you could use 5 or 10 minutes chasing one. And a slightly more adult juvenile gurnard, but again, it's beginning to grow these parts of its pectoral fin with outgrowths from that they use to sense what they're eating or looking for. And then to Connemara, to the clingfish, and your previous speaker, Joe Fitzgibbon, is really the expert on these, and I would have I sometimes refer to the next group of fishes when I come up from a dive, saying that I've seen some Joe fish, and everybody around that we dive with knows what I mean. So this one is the Connemara clingfish, and basically the clingfish have modified pelvic fins, and the pelvic fins are modified as a little sucker that they can stick onto the rocks underneath. And this one has that's the classic colours of that species. And then you get into the other clingfish. This one is the small-headed clingfish. And notice the colours a minute, blue spots with this prominent white, but there are all sorts of variations of it. Same species. And sometimes we see more of them than others. Generally, they're within the red algae, and you have to go sorting and patience looking for them. And then there's there's a sea snail, which also has this sucker on the ventral side, and they're called sea snails. Liparus is the scientific name, and they come in all sorts of shades. The eye is in focus, but the rest of the animal really isn't. But you get the idea of the stripes. And this was in July this year, and this seems to be a young one, not necessarily of this one, but nearby. So you get the idea of the variety of them. And you can get to see red mullet. These are in Lochine. And this year, and I didn't put it in, there was a baby red mullet in Newquay near the boat as you come out, near Jerry's boat. And then going back some years, this picture was by Joe of this large fish here, and it's a large fish. It's related to the sea snails that I was mentioning, even though this is a much bigger fish. So you can see that's me. This photograph was taken by Joe Fitzgibbon, and these are the pink eggs that I will show you in a moment. This is the male of the lumpsucker. That's it. That's it again. Huge fish, blob of fish with a large number of, so that's the previous ones. All of the shots of the fish are of males because the males are incubating the eggs. Unfortunately, that one was injured, and I don't think we've seen a lumpsucker. I'm sort of looking at Joe's face to see what he says, but I don't think we've seen lumpsuckers since in Newquay. And that was the male over the pink eggs. And that was its injury. So I basically want to say long, slow drives. And I'm thanking dive buddies, Tom MacDonald, Tony O'Callaghan, Joe, Benny, Frank, and Agret. These are the people who have taken me to these sort of interesting places. So thank you, and I'll take questions if there are any. Okay. Thanks, Tony.
Thank you. Thank you, John. I'm going to stop recording now just in case people would ask nasty